quality control test for the parenteral product. Means once your parenteral products are ready, that is ampoules, vials, infusion bottles, whatever it may be. Once the parenteral products are ready, now this parenteral product must be evaluated for various quality control tests. And what are these quality control tests? These are namely, first one is the sterility test, second one is clarity test, third one is the liquors test, and last one is the pyrogen test. So these are the three main quality control tests for the parenteral preparation. First one is the sterility test. This is related to the presence of microorganism, whether there is presence of any viable microorganism in your parenteral or not. That we can confirm by using the sterility test. And the sterility test must be always negative. Negative means there is no presence of any viable microorganisms. Second one is the clarity test. So in this test we are going to check the presence of any particulate material. And when you after visual inspection of ampoules or vials, there should not be any visible particle. If a single particle is present, then your test for clarity will be disqualified. The next one is the liquors test. Liquors test means after sealing of your ampoules, sometimes there may be fine cracks on your ampoules after sealing. Sometime after tip sealing, the ampoules may not properly sealed. There might be fine holes or fine cracks in your ampoules. So if there is presence of any fine cracks, so that can identified by using the liquors test. And last one is the pyrogen test. So what are the pyrogens? These are the metabolic product of the microorganisms. So if there is any presence of pyrogen, and this type of pyrogen containing preparations are if injected to any patient, there may be a rise in body temperature of the patient, or patient may be suffering from the body pain. So that's why we have to check the final product for this four quality control test, that is sterility, clarity, liquors, and the pyrogen test. So let us discuss one by one. First one is the sterility test. It is the method carried out to confirm the presence of any viable form of microbes in the product. Once your ampoules or vials are ready, then we have to confirm that the product is completely free from microorganism. And how to check whether the microorganisms presence or not? There are two methods. First one is the direct transfer method, and second one is the membrane filtration method. So by these two methods we can confirm the presence or absence of any microorganisms. Let us discuss first one, that is direct transfer method. So in this method we draw the required amount of sample. If you are having any ampoules or vials, whatever it may be, take the required amount of sample. You can see in this image we are going to insert the needle into a vial, and we are going to take the required amount of sample. Then this required amount of sample is transferred into two test tubes. First one, that is one half of the sample transferred to first test tube, which will contain fluid thioglycolate medium FTM. So first of half of the injection transferred to the fluid thioglycolate medium, and the another half transferred to the soybean casein medium. So in this way we are going to transfer the parenteral preparation in both of the test tube, fluid thioglycolate and soybean casein medium. The volume of the medium must be sufficient to promote and expedite the microbial growth. We have to add the sufficient quantity of your parenteral preparations, because these parenteral preparations are terminally sterilized, or it may contain some antimicrobial agent. So if there is a presence of any single microorganism, the small quantity of sample may not give the sufficient result. So in order to get the desired effect, you have to take the larger quantity of parenteral preparations. You can say near about 4 to 5 ml of the sample requires to get the desired results. And adequate mixing between the inoculum and the culture medium must take place to maximize the interaction. Adequate mixing is very much important between the inoculum. Inoculum means your sample, whatever the ampoule or vials you are taking, there must be adequate mixing in your sample and the culture medium. And which is the culture medium? We are using two types of culture medium, that is fluid thioglycolate medium and soybean casein medium. So once you are adding the injection in both of this medium, you have to do the adequate mixing. You have to adequately mix your sample along with the culture medium. Once the mixing is done, then we have to transfer these tubes into the uh, then we have to keep this into a inoculator, so that the inoculator will promote the growth of microorganism. So inoculator will provide the sufficient environment for the growth of microorganism. If there is a presence of any single or lots of microorganism, we are going to provide the desired temperature. And what is the desired temperature? It is about 37 degrees Celsius. So this 37 degrees Celsius temperature is very good, or it is favorable for the growth of microorganism. So once the adequate mixing is done in fluid thioglycolate and soybean casein medium, then we have to transfer this both of the test tube into the inoculator. Then inoculator will provide the sufficient environment and microorganism will start growing. And we have to keep this both of this test tube for minimum 7 days. Usually when we are going to transfer the E. coli bacteria or any Salmonella typhi or any type of bacteria into a culture medium, 24 hours are minimum. Within 24 hours you can observe the maximum growth of microorganism, because we are going to transfer millions of bacteria using a single inoculum needle. At that time we are sure that we are transferring the sufficient microorganism onto the culture medium. But here in this case, the microorganism may be present one or two or maybe up to 100, means the quantity is not sufficient so that within 24 hours you will get the desired effect. Means suppose one or two bacteria if present, suppose one or two bacteria accidentally enter into your parenteral preparation, so after 24 hours the growth will not be sufficient, and after 24 hours you will say that the test is negative, means the parenteral preparation does not contain any microbial growth. So why you can't see any microbial growth in 24 hours? Because the number of bacteria are very least. So that's why in order to check the results whether it is actually positive or negative, we have to keep this both of the test tube for minimum 7 days. So minimum 7 days you have to keep this both of the test tube in incubator, and after 7 days you have to check the result. Second one is the membrane filtration method. And it is for oil and oily preparation, alcoholic preparation, and for preparation miscible with or soluble in aqueous or oily solvents. Steps involved in membrane filtration are: the filter unit must be properly assembled with the must be sterilized proper prior to the use. So here in this image, left side corner, you can see a filter unit, and this filter unit must be sterilized prior to the use. Here you can see at the top side this filter assembly consisting of two glass unit, and in between this glass unit we have to keep our membrane filter. Once the membrane filter is fixed, then we have to close both of this glass unit by using this clamp, and once the clamp is fitted, then we are ready for membrane filtration. So before using this membrane filtration, first we have to sterilize this membrane filtration unit in the autoclave or by means of hot air oven. Once it is sterilized, then we can use for the membrane filtration method. Then contents are transferred to the filter assembly under strict aseptic condition. Once the filter unit is sterilized, once this filter unit is sterilized, directly you have to transfer this unit into the aseptic cabinet. And in the aseptic cabinet we have to filter the given solution. Suppose we are having the mixture of some ampoule or vials or infusion bottle, then this contents are transferred through this unit only in aseptic cabinet. Here you can see, first we have to fix the filter unit. Once the fixing is done, then we have to start adding the parenteral preparation, whatever it may be. Suppose it is in vials or ampoules, whatever it is, then we have to start transferring the preparation. After filtration, you have to remove the membrane filter and divide it into two parts. The membrane filter is removed aseptically and membrane is cut in half. One half is placed in suitable volume of fluid thioglycolate medium and another in equal volume of soybean casein medium. So in this way, once the filtration process is complete, then remove the filter, cut it in half, one half transferred to fluid thioglycolate and another half transferred to the soybean casein media. Interpretation of the result. So after 7 days, how you can interpret the result? If no visible evidence of microbial growth, then product complies the test for sterility. If there is no microbial growth, then it will comply the test for sterility. So here you can see a test tube which contains the fluid thioglycolate medium. So if the test tube remains clear, if the test tube remains clear, it means that there is no evidence of microbial growth, means it complies the test. If there is no microbial growth, we can say the test is negative, and this is only one test which we are expecting negative, otherwise all tests we are expecting positive. If suppose you are expecting there must be a red color in your chemical test, if the red color is there, then we can say that the test is positive for XYZ compound. But this is microbial test or the sterility test is only one which we are always expecting negative. Why we are expecting negative? Negative stands for no microbial growth. If the test is positive, it means that there is microbial growth and it will not comply the test. So if the fluid thioglycolate medium or soybean casein medium remains clear after 7 days, it means the test complies. And if microbial growth is found, it will not comply the test for sterility. Here in this image you can see different patterns of the microbial growth. If this type of growth is observed, if there is any turbidity or this type of cloudy nature in your test tube, that it means that there is a microbial growth in your preparation, and this microbial growth indicates a positive test, and that's why this does not comply the test for sterility. So this is all about the first test, that is your sterility testing. And the second one is the clarity test. This clarity test is for detection of the particulate material. The particulate matter in parenteral preparation has been recognized as an acceptable up to certain limit. Since the user could be expected to conclude that the presence of visible particle suggests that the product of inferior quality. So if there is any presence of visible particle, it means that the product is of inferior quality. As we are using this preparation for parenteral administration, means either IV or intramuscular administration. So this type of preparation must always free from particulate material, because if the particle size is more than 4 micrometer, then there might be chances for blockage of the blood capillaries. Once the capillaries block, it will not supply blood to the next organ or part of your body or tissues. So that's why parenteral preparation must be always free from visible particle. And how to detect the presence of visible particle? Entire product should be visually inspected by the human inspector under good light, against the reflection into eye and against black and white background. So this type of parenteral product are inspected by the inspector in the industries, and he can visually observe this product against black and white background. Dark background detects the presence of white particle and white background detects the presence of dark particle, and any container with the visible particle if seen can be discarded. Here in this image you can see how to detect the presence of white and black particles. So if the in this image you can see one person is observing the ampoules against the white background. So if we are observing the container against the black background, it means that we are trying to find out the presence of any dark particle in your preparation. So white background generally used for the dark particle, and dark background generally used for the presence of white particle. You cannot observe white particle against the white background. If you are going to observe these ampoules against the white background, you will not able to see any white particles. So that's why we have to use the contrast background. Here in this image you can see if there is a presence of any white particle, it must be observed against the black background, and if there is presence of any black particle, it must be observed against the white background. So in this way we have to perform the clarity test for presence of any particulate material. Second method of is the counter method for detecting the presence of particulate material. So how to detect the material by means of counter method? So in this case there will be increase in resistance as the particle approaches and passes through the orifice. So in short, if there is increase in resistance, it means that there is presence of particle. The particles are allowed to transfer in between two electrodes, positive and negative. If there is any particle, there will be increase in resistance. So this increase in resistance you can directly observe from the digital indicator. So if there is any increase in resistance, it means there is presence of particulate material. This method requires the destruction of product since an electrolyte is added to the preparation before its evaluation. So in this case, if you want to evaluate the ampoule, first we have to break the ampoule, and because we have to add some electrolyte in order to check this counter method. Some other methods are also useful for the clarity testing, and these are filtration method, scattering method, light absorption, and light blockage methods. Once the particles are detected, then they are identified by the various methods like microscopy, X-ray power diffraction, mass microscopy, microchemical test, polarized light microscopy, and scaling electron microscopy. So in this way we can detect the particles by using counter method. Third test is the Likers test. Means in this test we have to check whether the ampoules are leak or not, because most of the time after sealing of the ampoules there may be formation of fine cracks or fine holes in your ampoules. So if there is presence of any fine holes or fine cracks, there it may be responsible for the microbial contamination. So that's why before dispensing this type of containers into the market, first we have to check the check for the presence of any leakage. And how to check the leakage? And this Likers test is used to detect the incompletely sealed ampoules, so that they can be discarded in order to maintain the sterile condition. Once the leakage is detected, that ampoules will be directly discarded. Open capillaries or cracks at the point of seal results in the leakers. Whatever the cracks are present in your ampoules, it will be responsible for the leakage. And how to perform the Likers test? It is performed by immersing the ampoules in a dye solution which is made up of 1% methylene blue. Then apply the pressure of 25 inches of vacuum and for minimum of 15 minutes. So in 1% of methylene solution we have to immerse our ampoules and apply the pressure of 25 inches of vacuum and for minimum of 15 minutes. So after 15 minutes you can see this type of results. So in this image you can see that three ampoules are clear and three ampoules containing the methylene blue. So because of the pressure 25 inches of vacuum pressure, because of this pressure methylene blue will start to penetrate inside the ampoules if there is a presence of fine crack or holes. So through these fine cracks methylene blue solution may enter into your ampoules and whole preparation will becomes blue in color. So whatever the ampoules become blue, that will be directly discarded, means it is having leakage problem. Detection of liquor is prominent when ampoules are immersed in bath of dye during autoclaving. So this can be better performed by using the autoclaving. Why? Because in autoclave you can perform liquor test as well as the sterilization test. Both the tests can perform at a time when we are going for the liquor test using the autoclaving. How we can perform liquor test and sterilization? Because for the liquor test you require the pressure of 25 inches of vacuum, and that can be achieved very well by using the autoclaving. So in autoclaving you can perform both the tests, liquor test and the sterilization test. Bottles and vials are not subjected to liquor test because bottles and vials containing the rubber closure, and this rubber closure becomes flexible after keeping in autoclaving at 121 degrees Celsius for 20 minutes. So that's why this test is not applicable for bottles and vials. And the last one is pyrogen test. So pyrogens are the metabolic product of microbes, and these pyrogens are produced by most of the bacteria, molds and the viruses. So most potent pyrogenic substances are called as endotoxins, and they are produced by the gram negative bacteria. So the pyrogens are the metabolic product of microbes which are produced by the various bacteria, molds and the viruses, and the potent pyrogenic substances are called as the endotoxin, and this type of endotoxins are produced mainly by the gram negative bacteria. And when these endotoxins are injected into any patient, you can see a marked rise in body temperature, chills or body aches, cutaneous vasoconstriction and increased arterial blood pressure. So this is the effect of presence of endotoxins. So that's why your preparation must be free from pyrogens, because these pyrogens, potent pyrogens are called as endotoxin, and if this endotoxin enters in our body, there may be a marked rise in body temperature or the body aches also. So that's why all parenteral preparation must be free from pyrogens. And most likely the source of pyrogens are water, contaminated solutes and the container. So that's why always we have to use the sterile water for injection, and always whatever the solutes and container you want to utilize, everything must be sterilized prior to the use. Test involves the measurement of rise in body temperature of rabbit. So this test, pyrogen test, is mainly performed by using the rabbits, following the IV injection of sterile solution into the ear vein of the rabbit. So this pyrogen test can be performed by injecting the parenteral preparation into the vein of a rabbit, and then we have to check whether there is any rise in body temperature of rabbit or not. And how much dose we have to administer? Not exceeding 10 ml per kg. And which animals we can select? Only those rabbits which are healthy and adults and having the weight of not less than 1.5 kg. And what are the equipments and materials required? We require glasswares, syringe and needles to perform this test. Retaining boxes are also always required, because these are compatible for the rabbits as possible. And lastly, thermometers having the precision of 0.1 degree Celsius. So these are the requirements to perform the pyrogen testing. So preliminary test. Before going for the test, first we have to go for preliminary test, and which is also called as sham test. If animals are used for the first time or have not been used during the two previous use or 2 previous weeks, which animals we can use? If the animals used for the first time, means or those have not been used before 2 weeks, so this type of animals we are using for the preliminary test, and condition them for 1 to 3 days before testing. Condition them means we have to keep injecting 10 ml of IV preparation, pyrogen free saline solution, and whatever the solution we are injecting, that must be warm to about 38.5 degrees Celsius. So what is the first preliminary requirement? First we need the animals which are not used before 2 weeks or we are using for the first time. This is the first requirement. And the second requirement is that once the animal is selected, then we have to condition them for 1 to 3 days. Condition them for 1 to 3 days means we have to keep injecting IV 10 ml per kg pyrogen free saline solution. This is the first requirement. Then record the temperature of animal beginning at least 90 minutes before injection. Means before injecting, 90 minutes before we have to check the body temperature of rabbit, and then continuing for 3 hours after injection. Any animal showing a temperature variation of 0.6 degrees Celsius or more must not be used in the main test. After injecting pyrogen free saline solution, the body temperature should not rise more than 0.6 degrees Celsius. If there is a rise of temperature more than 0.6 degrees Celsius in any animal, that animal will be discarded. That animal we will not use for the main test. Then what is the main test? Main test is carried out by using a group of 3 rabbits. As you can see in this image, these rabbits are kept in a cabinet which are comfortable to them, and they will allow us to record the temperature very easily. Dissolve the substance in or dilute with pyrogen free saline solution. Whatever the drug you want to check whether it is sterile or not, that substance will be mixed with the pyrogen free saline solution, and warm the liquid to about 38.5 degrees Celsius before injection. Once it is warm, then inject the solution into the marginal vein of ear of each rabbit. Here in this image you can see marginal vein. So in this marginal vein we have to inject the parenteral preparation, and then record the temperature of each animal at half hourly intervals for 3 hours after injection. Means once the injection is done at 11 o'clock, then after 30 minutes each we have to record the temperature of each and every animal. This will be continue for up to 3 hours. Means in 3 hours you will be having 6 temperature reading of each rabbits. In this image you can see a group of 3 animal, and we have to record the temperature of each animal, and the temperature should be rising body temperature of each animal must be less than 0.6 degrees Celsius. Preparation being examined passes the test. If the rise in body temperature is less than 0.6 degrees Celsius, then it will comprise the test for pyrogen testing. If the temperature of 3 rabbit is more than 0.6 degrees Celsius, if the rise in body temperature is more than 0.6 degrees Celsius, then again we have to take a group of 5 animal, means 3 plus 5. In this way this test will be carried out in total 8 animals. And what should be the reading? Just look at this observation table. In first test, if we are taking a group of 3 rabbit, individual body temperature shall not be more than 0.6 degrees Celsius, and temperature rise in group of 3 it should not be more than 1.4 degrees Celsius. Then it will passes the test for pyrogen. Individual temperature 0.6 and group temperature 1.4. If any one of this rabbit does not comply the test, then we are going to take another group of rabbit, that is 5 rabbits. So total will be 3 plus 5, 8 rabbits. So in this case again individual body temperature may not be more than 0.6 degrees Celsius, and the group temperature should not be more than 3.7 degrees Celsius. If it complies this test, then it will passes the test for pyrogen. And if above test not passes, then sample said to be pyrogenic. Means if the reading is more than 1.4 or 3.7, then the sample is said to be pyrogenic, means it contains the pyrogen, that is toxic endotoxins. Next one is the bacterial endotoxin test or the Limulus amoebocyte lysate test, that is also called as LAL test. The bacterial endotoxin test is the test to detect or quantify the endotoxins from gram-negative bacteria using amoebocyte lysate from the horseshoe crab. So, as earlier we have seen in sham test and uh, main test, in that we have seen that the toxic pyrogens are called as the endotoxin, and these endotoxins are mainly produced by the gram negative bacteria. So this is the another test which can detect the presence of bacterial endotoxin, that is bacterial endotoxin test, or which is also called as LAL test. So how we can detect the presence of endotoxin? In this case, we can detect the presence of endotoxin by using amoebocyte lysate. So this is the enzyme or proteins which are secreted by the horseshoe crab. Here in this horseshoe crab is one of the uh, sea animal. So from this horseshoe crab we can obtain the enzyme that is called as amoebocyte lysate. If there is any presence of endotoxin, this amoebocyte lysate will react with the endotoxin, and then there will be formation of gel or precipitate. So if there is formation of any gel or precipitate, it means there is a presence of endotoxins in your preparation. The endotoxins of the gram-negative bacteria forms a firm gel within 60 minutes in presence of lysate of amoebocyte. So within 60 minutes there will be formation of a gel if there is a presence of endotoxins. Hence, a test is only effective with the gram-negative bacteria, which constitute the majority and most potent of the pyrogens. The addition of solution containing endotoxin to a solution of lysate produces the turbidity, precipitation, or gelation of the mixture. Means once the enzyme obtained from this horseshoe crab it is mixed with the endotoxin, there will be formation of turbidity, precipitate, or gel type of structure. So this type of turbidity, precipitation, or gelation is the confirmatory test for the presence of endotoxins. Here in this image you can see we are having one horseshoe crab. Why it is called as horseshoe crab? If you see the structure of this horseshoe crab, it is just exactly similar to the horseshoe. That's why it is called as horseshoe crab. And from this horseshoe crab we are going to obtain the enzyme and proteins which is called as amoebocyte lysate. And when this amoebocyte lysate mix with the endotoxin, within 30 minutes there will be formation of gel, and if there is formation of gel, your product will not comply the test for endotoxins.